Hi guys. So let me get everything set up for this. Sorry, I was sending out an email and got a little delayed with that. So we're gonna go over chapters 17 and 18 today. I also wanted to add before we get started, I'm gonna go through the Unit 3 review questions that we're gonna be doing the Kahoot for. I already sent out the link to the Kahoot for you guys to start practicing on and studying with. I'm going to take some time. I've been so busy this week dealing with some crises that have to do with the faculty side of things. I haven't had time to really sit down and nitpick on the questions on the Unit 3 Kahoot. So I am going to be taking time today and tomorrow to refine those questions so that I can assure that they are addressing topics that I will expect you to know at that detail for the exam. They will not be the test questions. They will not be comprehensive over every single thing, every little detail. But I will try to get great representation for the level of understanding that I am expecting on the topics that could be addressed as much as possible. So I will send out an updated version of the Unit 3, or at least like email out an announcement saying that I have updated that once I get those updated. But I wanted to let you know that I will be working on updates on the Unit 3 Kahoot. I don't know if it'll be major changes. I don't think it'll be like all the questions changing, but I just want to ensure that you guys understand the level of understanding that I am expecting out of you guys for the exam. Because there was question about that, and I've had a lot of emails like, you're not going to actually ask us, for the type of hypersensitivities, you're not going to ask us that type 1 has to do with IgE, right? And I'm like, so I'm not going to tell you that I'm going to ask you that question on the test or not, but I do expect you to actually know that and be prepared for questions like that. And so I want to set up the Unit 3 Kahoot to match my level of expectation as it relates to the Unit 3 review, like the Unit 3 study sheet and the content on the study sheet. And so I'm going to fine tune that Unit 3 review questions Kahoot to better reflect that so that people, because I know everybody pretty much leans on that. I also want to reiterate, I very, very strongly disagree with giving out exam questions and exam answers, period. That does not facilitate learning. It facilitates word regurgitation, and that doesn't help with understanding the material and no matter what program you guys are going into, they're expecting you guys to understand this material, and they're going to start delivering those classes pretty much immediately with regard to assuming you understand that material, not that you were able to regurgitate for this question in this particular instance. So we're trying as a whole, the science department is, to make sure that our classes are actually designed in that regard. So blah, blah, blah. Let's move on with what we're doing today. Let's pull up my slides. I need to do share. Where there it goes. Share screen. Start broadcast. Okay. All right. Let's do it. I do want to, let me bring up the chat really quick. Chat. There it is. Let's split screen. Put this over, and then chat over here in case you guys have questions. I can at least see this stuff. All right, so we have Heather. The Unit 3 study sheet made by me didn't have osmotic pressure on it under Chapter 11. Okay, so it used to, and so it is not now, and you will not have osmotic pressure on the test. I can verify that. We did cover it in the lecture, and it used to be on the study sheet, and I took it off recently for this semester. This is the first semester I took it off, and I hadn't updated the slides. So you're right on that, and that will not be on the test either. I can assure you on that. So thank you for bringing that up, and don't worry about it, is what I'll say. So that is fair. That's a fair point. But yes, I have tailored the exam to what's on your study sheet. And yes, I know. I'm sorry. Yeah. But I always tailor the exams to what is on the study sheet. And if it says, so another question that some students had, um, the study sheet, it says like in phenotypic, it has the culturing methods, and it says in there selective and differential media. We have talked in previous chapters about what selective and differential media do. We learned in chapter 3 and in the lab that differential media will have changes and usually based on color changes in the media or the growth on the media due to metabolic byproducts, right? So differential media is a metabolic or biochemical test that does result in color changes, and that makes it differential media. The fact that it relies on metabolic changes makes it biochemical. That's a biochemical test, and then biochemical tests all fall under phenotypic. And so when we ask about a question of if you use media that has metabolic change that causes a color change in the media, what type of test is this? That means it is, if it's a media that's doing that, it is differential media. If it is a biochemical test, then it is biochemical. And if it is biochemical test, it falls under phenotypic. So it is all three, but it's about understanding those subcategories on that as well. And that if the study sheet says selective and differential media, you don't just need to know the word differential media. You need to know what differential media is, because we have talked about it in previous chapters. I didn't define it in that study sheet because the other study sheet for unit 1 defined it, as well as the lab defined it. And so if there are statements on there, like when you look on the study sheet and it says immunologic tests for chapter 18, and under immunologic, it says, or immunology tests, it says serology. It just says serology. If you don't know what that word means, I didn't define it in the study sheet. The study sheet doesn't contain everything you need to know about the topics. It tells you about the topics that I could test you on. And so if it just says serology, it's on you guys to understand what serology is if you don't know what that is. And so be aware of things like that in the study sheet. The study sheet is, for all intents and purposes, the study sheet is learning objectives. Those are the topics that you should have learned and understood. Yeah. So definitely that's why I always push for exactly that, Heather, which is to say that you should take the study sheets and be able to explain to me everything on the study sheets and what they mean and everything. And that's why taking the notes based on the study sheets is essential. Those are your learning objectives for my course for each chapter, learning objectives for each chapter. So this says week 12 and I updated this to say 17. No, that's right. It's week 12, chapter 7. Right, never mind. Oh, it's so much, you guys. It's so much. It's a lot for me too. It's a lot for me too. Okay. Let's get going. Before I get started with the week 12 stuff for chapter 17 and 18 here, does anybody have any questions about this test? Shouldn't be different than the other tests in that regard. Like I'm still expecting you to learn the study guide, and I'm still going to test you based on the study guide. The test hasn't changed in that regard. It just has only changed the actual questions on the test. And that is specifically because the test questions and their answers, the entire bank got leaked, not just from my class, but for other classes. And so if you were relying, and I'll say it again, I said it in the classes, but I'm gonna say it one more time. Yes, same exact concepts, just different wording. And so be sure that you understand the concepts that are covered on the study sheets that I gave you and the study guide that you may or may not have gotten from STEM lab from the SSOC if you got that. Those are still the concepts that I'll be testing. It's just that the study sheet that the STEM lab gave out was my literal verbatim word for word questions and their answers. That is unacceptable. I will not have students memorizing my questions and the answer and just using word association. So that's why the questions on the test are going to change, but they're still like the similar concepts. If you understand the concepts on the study sheet that I gave you, if you understand the concepts that those questions on the study guide that the STEM lab gave out, you understand those concepts, you should be just as fine as any other test. And if you were a student that was studying the concepts, This should not have changed anything for you. If you were a student that was memorizing the study guides from the STEM lab, that is the only situation that should have changed. So I just want to make that clear. Because a lot of people are like, you're changing the test, you're making it harder. No, I am not. I am not, I promise. I am just changing the wording. I'm still testing the same exact concepts. I hope that's clear. Really just needed to, we don't know how long this has been going on. I'm sure students would be able to tell me how long this is going on. But we don't know. So the only way to fix this was to change the test, and there's really just no other approach to it. And if you are taking anatomy or physiology, the same thing. It's just the same thing. So if you got study guides from STEM Lab, from anatomy or physiology, no matter who your professor was, again, they are updating their test banks. So do not rely on the questions and the answers being the same as the study sheets that you got from the STEM Lab. FYI, probably odds are if you're joining me on this conversation, you're probably not one of those people. I'm just saying, you probably were studying the concepts, and kudos to all of you guys that were doing that. But you are also probably aware that there are plenty of students that were just memorizing the study guides. And we're putting a stop to that. Does anybody have any questions that's on here? Does anybody have any questions regarding that stuff? Make sure that my volume is up on this. I'll unmute that, just in case anybody had any questions. If not, we're going to go ahead and get started with week 12. So this will be chapter 17 and 18 review. And on Sunday, again, we will use the Unit 3 Kahoot game as a game, guide, and we'll play the different games with those questions. And I will update them in the next 2 days to make sure that they are covering the content at the level that I expect you to understand them wherever possible. I think it's pretty good as it is, but I want to be sure that there are some details that you understand. You need to know the details. Cool. Okay, let's get into it. Let's start with what we're going to cover. We're going to talk about chapter 17, whatever this is doing, I don't know. Chapter 17, and then 18, then the most missed topics for each of those chapters. So for chapter 17, this was our, immunopathologies. These are the diseases of the immune system. When you have hypersensitivity, we are reacting to something that you're not supposed to react to, as it's not a pathogen. So that's what hypersensitivities are about. Immunodeficiencies are whenever you are not reacting to a pathogen at the level that you should be. So there's those concepts there. Let me see. Oh, I did not mean to click that. Whatever. All right. So our hypersensitivity groups, those, how do I put this in actual, oh, this is just going to take the whole screen, isn't it, if I do that? Whatever. Okay. So our hypersensitivities, type 1 over here, this is going to be based on type 1 IgE. And yes, you might be tested on which immune type reaction is associated with each hypersensitivity type. So type 1 hypersensitivity, these are guided by IgE. And when we look in our study guide, what actually happens is mast cells use IgE as a receptor. So your B cells will make IgE against the allergen. The IgE circulates. It gets worn around by the mast cells as a new receptor on the surface of the mast cells. And those mast cells can have those IgE molecules as a receptor on their surface for decades. And so anytime that you come into contact with an allergen, the allergen acts as an antigen that binds to the IgE molecules. And when that happens, that causes the mast cells to degranulate. That's what's actually happening there. The mast cells, when they degranulate, send signals out to the body that are the inflammatory response, basically. And that can be atopic, which just means local, version, and this includes allergies, typical allergies like hay fever, like asthma, like eczema. So those are all local responses due to IgE causing mast cell degranulation. Then we have anaphylactic shock, which is a systemic response. And so anytime the signals are being sent through the bloodstream and throughout the body that way, we get an anaphylactic shock response. These responses can be triggered by inhalants that you breathe in, contactants that you're actually surface to surface contact, injectants where we have an allergen being injected into the body or into the bloodstream, and then ingestants where you are actually getting it through eating or drinking, ingesting it into like the GI tract. All right. All of these responses require an initial sensitization, just like when I got the chickenpox. So when I got the chickenpox, that was my initial sensitization to the chickenpox virus, which is varicella zoster. And so when I saw that virus, it took my body a week or so to build up my antibodies against the virus. And those antibodies, as well as my cytotoxic T cell response, helped clear out the virus. Now my body has memory to it. So the second time and every time thereafter, which we call the secondary response or the amnesiatic response, every time that I see it after that, I will respond instantly. So now when I see the chickenpox virus, my body immediately has memory and can recall that immune response immediately. And so allergy is your body recalling your immune response immediately, but it required an initial sensitization. So that meant at some point I saw a cat for the first time and I didn't have an allergic response the first time. My body was creating that immune response, treating the cat dander as an allergen, and it took it a while to build up IgE. Every time after that I see a cat, I will immediately have the IgE response and the degranulation of the mast cells. So that initial sensitization, that's what that is. So IgE is that mast cell receptor after that point, and we'll see the allergen and it degranulates, releasing things like histamine. And that so in order to treat our allergies, we can use things like antihistamines, which are very effective for treating local atopic allergies, or epinephrine, which is used to treat our systemic allergies. All right. We can also get rid of allergies by triggering IgG production against the allergen. And so IgG will bind with the allergen and just normal IgG response. And so when your IgG sees chickenpox, you don't get chickenpox symptoms. You don't get inflammatory symptoms. You don't get itchy eyes. You don't get hives. You just get your IgG response just eliminates it. And so instead of you having an IgE response leading to the itchy eyes, the hives, etc., you get IgG to just eliminate the allergen. And so IgE never gets a chance to have the allergic response. Yes, I can do that. The only problem with this is that if you guys do chat on me when I'm doing my talk, the only problem with that is going to be that I can't see that you're chatting with me. So FYI, if you have something to say, I'll close the chat box. I'm going to do that. But if you have something to say, open a mic or something like that and let me know, because it doesn't pop up with the chat during the thing. Just FYI. So I will do that. Let me close this. Hold on. Let me do this full screen. All right. Let me actually open this as, since we're doing that, let me try to open this as a presentation. Well, I'm trying. This? No. This is what I want. Okay. All right. There we go. That's better. Okay. So That's the type 1 hypersensitivities. Those are the allergy responses related to IgE. IgE causes binds on the mast cells. The mast cells, when the IgE on the mast cell sees the allergen, that mast cells degranulate. That causes the inflammatory response. And if you initiate IgG, IgG will bind up the allergen so that the IgE and the mast cell response can't happen. So you'll just remove the allergen instead. Okay, type 2. So now we have red pen instead, I guess. So type 2. So type 2 is mediated by your IgG and IgM. Your typical response to, if you had a pathogen, you would be making, your main response would be IgM and IgG. And those are, IgG is important for the memory stuff. And what happens is the IgG or the IgM that is against the other blood types than you have. If your body sees that blood type as foreign, it will make IgG bind to the red blood cells with the other marker on it, the foreign marker. And then that initiates the complement cascade based on the classical pathway of complement activation. And then this will cause the complement to create that MAC, membrane attack complex, that pokes holes, typically in pathogens, but it'll be poking holes in the red blood cells. This releases the contents of the red blood cells, including hemoglobin. And when that happens in your body goes into shock, and then that can lead to death. So this is a blood group incompatibility issue. Our 3 blood group markers are A, B, and O. Sorry. A, B, and O. If you have A or B in your genes, if you have even just one copy of A or B at all in your genes, you will express A or B. If you have A and B, you will express both A and B. And so AB blood types, they have a genetic type, you know, automatically they got an A and they got a B. And those are their 2 genes for that. For people that have an A blood type, they could have had a gene for A and a gene for A, one from the mother and one from the father, or they could have an A and an O. If you have A or B, you must express those as your blood type, both equally. But if you have O as one of your genes, O will only be your blood type if you have 2 O genes. So it is a recessive trait. And so that is what's going to dictate your blood type. Heads up. I'm explaining this so that you understand, because I get so many questions about genetic relation to blood type. Well, my dad was a B. How am I an A? Or something like that. I'm explaining this for your personal understanding. On the exam, I will not ask you about the genetic side. I will not ask you if your patient has a genetic A and O, what is their blood type? On the exam, I will ask you questions relating to the type 2 hypersensitivity stuff. So I will ask questions like, if your blood type is A, can you get type B blood? No, because A people will have anti-B antibodies. B people have anti-A because A looks foreign to them. If you are an AB, A looks normal to you, B looks normal to you. And then O is basically the absence of A, so A will look foreign to you, and the absence of B, so B will look foreign to you. So both look foreign to them, so they have antibodies against both. And so those are the things that I'll ask you about on the test. As a result of this, nobody ever gets anti-O antibodies because O is kind of the foundation for everybody. Everybody can receive O, and so O is the universal donor. O can donate to everybody. AB blood types can receive any ABO blood group. But blood types aren't just defined by the ABO blood groups. Blood is also defined by the rhesus factor, which is the RH factor. And I won't test you that RH test stands for rhesus. I won't test you on that. But definitely know that if you are Rh positive, like I am A positive, so I'm Rh positive because I'm A positive, that if somebody is Rh positive, that Rh, the fact that it is there, looks normal to them. If you lacked the Rh, that's not a problem either. It just means that it's just not there. But if you are Rh negative blood type, right? If you're an Rh negative blood type, you have never seen Rh factor before. That is foreign to your body. So people who are Rh negative cannot receive Rh positive blood. And a mother who is Rh negative will develop antibodies against her Rh positive baby's blood. This won't be an issue until there is a basically, I don't want to say rupture because that's not accurate, but there is a break in the placental barrier during the birthing process, that the placental barrier keeps that under control. But once the placental barrier is compromised in the birthing process, any antibodies that were made against that baby's blood type will be allowed to attack that baby's blood by crossing that placental barrier during the birthing process. And that leads to hemolytic disease of the newborn. And that's due to the Rh negative mother creating antibodies to the Rh positive baby. It does require sensitization. Same thing as me having the chicken pox. I had to build my immunity. My first exposure, when I got the disease, I didn't have immunity. My second exposure to chicken pox, I can fight it immediately. It's the same thing with a mother that is Rh negative with an Rh positive baby. The Rh negative mother with her first Rh positive baby was just developing her immunity. But the second Rh positive baby, her immunity can attack it immediately. And that's a sensitization process that is going to cause that. So that is type 2. Let's move on to type 3. These are based on immune complex. This usually takes large amounts of different antibody, and I usually focus on it being IgG. It doesn't matter what kind of antibody it is, but it's the fact that it's not just antibody, and that antibody is recognizing the antigen, like in type 1 and type 2. In type 1 and type 2, you have antibody, and when the antigen comes about, you get your reaction of either your allergy for type 1 or your blood rejection for type 2. In type 3, we have pre-existing antigen that is already bound to antibody, and those complexes get stuck in your blood vessels and a part of the blood vessel called the basement membrane. It could be blood vessel or it could be tissue. And that those complexes get stuck. Oh, geez. That was weird. Okay, sorry. That really surprised me. It took me off guard. Okay, so moving on. Where did I even put my pen and all that? Let me mute this. That doesn't happen again. Okay. Anyways, immune complex. I don't know if you guys even saw that. I got popped up with a phone call and it just like took over my whole screen. So anyways, immune complex. The fact that the antibody and antigen are already bound together and they're not being cleared by the immune system yet. And they get stuck in these very fine capillary areas in the basement membranes of your blood vessels. And they get stuck there, and the neutrophils keep attacking that, thinking that it's something foreign. And then that inflammation that the neutrophils are inciting causes issue in the tissues surrounding where that complex got stuck. And so while the body's trying to clear out antigen, it creates those large groupings of antibody and antigen called a complex. And then when they get stuck, the inflammatory response happens and it affects the tissues in the area. And this is what we see, the only one that will be on your test. Okay. Acute post-streptococcal glomerulonephritis is when you have streptococcal infection. Streptococcal infection would be like Streptococcus pyogenes, which is a species that causes strep throat. So if you have strep throat, and after your body is trying to clear it with your antibodies, some of those antibodies bind to some of the antigens from the strep bacteria, and those complexes get stuck in fine capillaries where we have gas exchange or elimination of gas exchange, like what we have in all of our capillaries, or elimination of toxins like what we have in the kidneys. And when this happens in the kidneys and it gets stuck in the kidneys where we're having elimination of toxins, the neutrophils cause inflammation in the kidneys. The part of the kidney where this happens is called the glomerulus. And you'll learn about the function of the glomerulus in your anatomy and physiology classes. And I'm not going to test you on that in my class, but it is part of the kidney. And then the inflammation that is caused by responding to those stuck complexes is where you'll get that inflammation that we call the itis. So glomerulonephritis means the glomerulus of the kidney is being inflamed, and that's from having had strep throat and leading to the immune complexes. And this is acute because it is only associated with recovering from strep throat. Your body will eventually eliminate it, but if you have enough of an inflammatory response in this particular situation, it can cause permanent kidney damage if it's strong enough. And it can also lead to the need to have temporary dialysis. And there are other effects that can be caused from having glomerulonephritis that could be more or less severe in that regard. So that's type 3. Type 4. These are our delayed type hypersensitivities. They are delayed because when you have a B cell that is a memory cell, the B memory cell might have its little receptor. Because it's seen the antigen before, that B memory cell doesn't have to go through the whole process of activation that we learned about in chapter 16. The memory cells, when they see the antigen come up, can immediately just start cranking out antibody immediately. So this would be IgG, which is our workhorse, found in your bloodstream in high concentrations. The IgG can go and eliminate the antigen and the pathogen associated with the antigen very, very quickly. And that's immediate. However, when you have cytotoxic T cells that are CD8 positive T cells, cytotoxic T cells that have memory, when these guys get activated on their little T cell receptors by MHC1, right? So this is presenting antigen. The T cell receptor sees it. We are a memory cell. We don't need to go through the whole process. This is just one cell or maybe a small handful of cells. That's not enough to get rid of an infection. So what has to happen is that these guys have to multiply first. The word that we keep using in our chapters is proliferate. This takes a couple days. This takes time. Then those proliferated cells will be actual functional cytotoxic T cells that can go kill infected self cells. So that memory response for your cytotoxic T cells, if you only had cytotoxic T cell memory and didn't also have your antibody memory, then every time that you saw a virus, if you only kept cytotoxic T cell response, every time that you re-saw a virus that you've seen previously, your memory would take a couple days to catch up. So you would be mildly sick for a couple days. You wouldn't be immediately prevented from getting sick. So that's why we have to have both in our memory. But in situations where this is a problem, where we have cytotoxic T cells that see cells as being infected with something when they're not, those situations are usually related to these. These are the obvious examples. The tuberculin PPD skin test. The tuberculin PPD skin test, we are injecting a little bit of tuberculin antigen into your skin. If you have cytotoxic T cell memory to tuberculin, a couple days later, after getting that small injection, you will have swelling and an inflammatory response for that after this has happened. Takes a couple days, right? That's because tuberculosis bacteria lives inside of cells. And so you will actually see your cytotoxic T cell response in that inflammatory area in your skin a couple days later because of that T cell memory if you have had tuberculosis and seen it before. If you haven't seen it before, you shouldn't be getting that response. Poison ivy and other contact dermatitis is driven by this response as well. And so those responses can take a couple days. If it's a delayed, takes a couple days event, it is the cytotoxic T cell response that is leading to that inflammation, not your antibody. Antibody is immediate after the initial sensitization. So anyways, the graft rejection operates on the same concept. All of these also require an initial sensitization. We were sensitized when we created our memory cell originally in our original response, which we covered in chapter 16. Now the memory cytotoxic T cell has been turned into a memory cell. It still takes it a couple of days to respond, but it's faster than the initial response, which took, 1 to 2 weeks typically. This is what drives our graft rejection in our transplants. And this could be either you get somebody's kidney put into your body and your body rejects the kidney. So if your cytotoxic T cells are attacking the kidney, that is the host attacking the graft. That is host versus graft. If you get a bone marrow transplant and the bone marrow cells contain T cells that are not yours, they are from somebody else. If those T cells see the host cells as foreign, they will attack them and think that they are infected cells. So this is the same exact reaction, except the bone marrow T cells are going to be attacking the self cells. That is the bone marrow graft attacking the host. And we only see that in bone marrow grafts. There are different kinds of grafts that you can get, and you will be tested on these terms on the exam. The autograft is when you get a tissue transplant from your own body. If you get a burn on your hand and you need tissue, it burns it so bad, it's a 3rd-degree burn, and you can't regrow your own skin in that area because you got a bad enough burn or it's a large enough area. We can take skin from another part of your body and then put it onto yourself. That's an autograft. An allograft is from genetically non-identical people. So this is different than the isograft. This is why I'm making this distinction here. An isograft is from an identical twin. Now, we don't clone people, but if in the future I can make a genetically identical clone to you, like in the movie The Island or something, right? They were literally like rich people made clones of themselves so they could harvest body parts from the clones. And so that's a potential thing where you would be genetically identical. But in reality, in the real world that we actually live in right now, at least according to what we are led to believe, I don't know, have your own conspiracy theories. But in the real world, your only time you're going to have a genetically identical person to you is your identical twin. Your sister that is older than you is never genetically identical to you. Your sister that is younger than you were not born as identical twins. If you are, what do they call them? Fraternal twins. You are not genetically identical twins. That you're not genetically identical. That is not an isograft. The only time isografts occur is when you are genetically absolutely 100% identical. And that's what you get with identical twins. Right now, that's the only scenario. However, if you are not genetically identical, that is an allograft. So from another person, no genetic match. So if I give a kidney to my little sister, that's a allograft. She's a little sister. She is younger. That gives me enough information to know she's not genetically identical. So on a test question, if you get asked a question, an older sister donates an organ to a younger sister, what type of graft is that? You should know it's an allograft. It's human to human, and you automatically know because they are not identical twins because they weren't born at the same time. And I didn't tell you they were identical, genetically identical. Then that is an allograft. Isograft is only going to be identical twins. A xenograft would be from non-person to a person. So cross species. We have heart valves from pigs. We have heart valves from cows, and other tissues that can be derived from other animals. Those are xenografts. Okay, moving on to autoimmunity. There are autoimmune diseases like lupus, rheumatoid arthritis, Graves' disease, and type 1 diabetes. That these are all driven by one or more of these hypersensitivities that led to anti-self immune action. And so one of these immune activities somehow drove attacking your own cells. Certain microbes might be able to mimic our molecules, and then that causes us to attack the microbe. And then it's similar to our cells, so we accidentally attack our cells because they look the same. It could also just be due to viral infections that can alter receptors on the surface of cells and make our receptors look different. Like it can make our receptors, they used to look like self. Now, because of the virus changed to our body, now our self molecules don't look like self anymore because of that. And we can also have just inflammation triggering accidentally the anti-self response. We can also have a change in our gut biome that can disrupt how our body trains what you should react to and at what capacity you should react to it. And all of these things can play into triggering autoimmunity. All right. Our immunodeficiencies, there's two major groups of immunodeficiencies. Primary immunodeficiencies are going to be genetic defects that are present from birth. They are innate. You are born with it. You didn't acquire it later on in life. So these are genetic defects. This includes things like severe combined immunodeficiency, where you have a genetic defect that inhibits your ability to create appropriate B cell and T cell responses. So you get no adaptive immunity. You never get memory to any infection ever, forever, for the rest of your life. These people that have this usually die very early on in life. And it's very hard to prevent infections with people that have severe combined immunodeficiency like that. Typically, it is a death sentence. You can live for years, but at some point, usually the infection is going to win. And you can get infected from very, very benign microbes because you don't have an immune response to protect you. The other type of immunodeficiency is called secondary immunodeficiency. It's secondary because you get it later in life. You always acquire it. It's acquired somehow. If you acquire it because of an HIV infection and the HIV infection wasn't controlled with medication, then HIV infection will always lead to the development of AIDS. And AIDS literally stands for acquired immunodeficiency syndrome. You have a low level of CD4 positive T helper cells. Those T helper cells are our captains. The T helper cells now can't activate your memory. Your captains cannot activate your B cells, and they cannot activate your cytotoxic T cells. So you can't have immune responses that lead to memory once you have the development of AIDS. So it's acquired due to HIV infection. You can acquire immunodeficiency if you have a cancer in your immune system. So lymphoma is going to be dealing with your lymphocytes. They're overgrowing, uncontrolled, and now they aren't functioning properly because we're not behaving the way that we're supposed to do. And so that can lead to an immunodeficiency from the lymphocyte side. Leukemia is a cancer that affects white blood cells in general. So you could have immunodeficiency because your white blood cells aren't doing their thing. You could also have chemotherapy or radiation to treat other cancers, even non-immune cancers like breast cancer or colon cancer, that the chemotherapy and the radiation will also kill off your white blood cells. That can be temporary or it can be permanent depending on the level of chemotherapy or radiation that you're receiving. But those are acquired due to those exposures. So that is our summary for chapter 17. We're going to move on to chapter 18 and summarize that after we get past this weird random blank slide. So chapter 18 was our diagnostic tests. We have 3 major groups of diagnostic tests. And so let me break them down. And this is organized a little differently than the study sheet, but we have all of these are phenotypic. All of these are immunologic. It is a major category, but we already discovered in previous chapters, we discussed PCR, we discussed nucleic acid hybridization, we discussed microarray. That was all in chapter 8. And so I didn't outline it here. So we don't have as much detail on it, but you still need to understand those details for those tests because we covered them previously. But this is genetic or genotypic, same thing. And they are the same thing. I will not give you a choice on the test between genotypic or genetic as the term for it. Promise. Okay. So those are those guys. And then these guys are in their own other category. So there's kind of 4, but the 4th one is other. They just don't fall into the top 3. Okay. So of course, first you have to collect your specimen. And there's all the different ways you could collect your specimen, from the saliva, from the blood, from the feces, from the urine, from sloughing of skin cells, from hair samples, or whatever it is that you're testing. And then you need to know which tests you're ordering. And if you're needing multiple tests, and you want to also consider, of course, like, is it necessary to use the expensive test when we could use the cheaper test and get the same answer or whatever? Those are the steps involved in deciding what test to perform. These guys here, we have the phenotypic, the immunologic, and the genotypic, and the other. For the phenotypic, if you are cultivating, so that means culturing. Cultivation means culturing. If you are culturing your pathogen and then using the results of the culturing process as part of the steps to determine what kind of organism it is, that's a phenotypic test. So here we're talking about isolation media, so that we can actually isolate the organism. That would be a step in collecting your sample. Then our selective and differential media. Selective media, it has ingredients to stop some things from growing and allow other things to grow. We learned in the lab that EMB is a media that inhibits gram positives and allows gram negatives to grow. So if you take your patient sample and you streak it on EMB and it grows, it's probably gram negative. But it takes it a step further to the differential side. So differential media usually is depending on metabolic waste products that lead to a color change in your media. Our entire rest of our semester in the lab is focusing on differential media. And it's all differential media based on biochemical changes from your bacteria. Does your organism ferment glucose? Does your organism ferment glucose and make gas also? These are all things that tell you information about the organism based on their metabolic or biochemical differences. Those are the same thing, by the way. And so their waste products that they make from those biochemical processes are causing a color change in the media. That color change is telling you that this biochemical process happened. The fact that this biochemical process happened or did not happen, and then that information is used to identify your organism, makes it a phenotypic test. So those are our biochemical tests, and we often will combine multiple tests in our phenotypic tests, not just one media. You grew on EMB and you turned it metallic green. Well, that might suggest that it might be E. coli, but that doesn't definitively say that. And so if you need more information, if you're not sure that it's E. coli, or you don't have any other evidence supporting that it might be E. coli, you might do other tests to support that. So an example would be starting with you know that this is a bacteria. Your first test might be the gram stain. Mess this up. Hold on. Oh gosh. Try to switch this to eraser and it's not letting me. There we go. Okay. So you have your bacteria and you are either gram positive or you are gram negative. If you are gram positive or gram negative, right? And so then based on if you're gram positive, we might be able to look at if you are a cocci or a bacilli. And then if you're a cocci, well, if you're a cocci, did you grow on mannitol salt agar? Just growing it on mannitol salt agar doesn't let you know that it is gram positive or that it's a cocci. But the fact that you're gram positive and the fact that you're a cocci and that you grew on mannitol salt agar and that you turned the mannitol salt agar yellow very highly suggests that you're probably Staphylococcus aureus. It's not just the fact that you turned mannitol salt agar yellow, but the fact that you were gram positive coccus, grew on mannitol salt agar, and turned it yellow, that all of those things, that plays into that concept of the dichotomous key. You may be thinking about these things and using them in the process without actually referring to a physical dichotomous key. That process of still thinking of if you are gram positive or gram negative, if you are a coccus or a bacillus, if you are positive or negative for endospores, if you are positive or negative for the acid fast test, if you grew on staphylococcus media or didn't grow on it, if you turned your phenol red fermentation glucose broth yellow, those things are taking all of that stuff into consideration, which is exactly what we're doing in the lab. That process of thinking of if you're positive or negative for this trait, that is the theory of the dichotomous key. All right. Antimicrobial sensitivity. You can actually basically make these, we call them abiograms, but essentially the identity for which antibiotics you are susceptible to and how susceptible you are to them can sometimes be used to identify bacterial species from one another. That's the idea of that. So we learned about the Kirby-Bauer method previously, but the fact that you have a really big zone of inhibition around ampicillin, if you are, Staphylococcus aureus, but it might be smaller if you are Streptococcus pyogenes, that can be used for identifying organisms from one another. Phage typing. We learned in chapter 5. We learned in chapter 7 that when we learned about viruses, phages are bacteriophages, are viruses that infect bacteria. Viruses that infect bacteria only infect one species of bacteria. Always. That's a hard and fast rule. So a virus that infects E. coli can only infect E. coli. And so we can use that knowledge to say, I think my patient has salmonella, take a culture from that stool sample, expose it to a virus that only infects salmonella, and if that virus infected your patient's sample, and we see that that virus infected your patient's sample, that indicates that your patient had salmonella. If the virus did not infect your patient's sample, your patient did not have salmonella. They had something else. We'll have to do other tests to determine what. So that's phage typing. Bacteriophages only infect one specific species of bacteria. For the immunologic tests, this is going to be based on antibody or antigen recognition. So it's not always antibody. It's not always antibody. It is not always antibody. It is based on immune response to antigen sometimes. That doesn't have to do with antibody sometimes. What am I trying to get at here? This is our bottom one, tuberculin skin test. This is an in vivo test. In vivo. Vivo refers to living tissue. Vitro refers to a test tube. Situ refers to tissue that is no longer attached to the living body anymore, but it's sort of in between. Okay. So we haven't broken down the cells. The cells are still intact in the biopsy. That's in situ. And in utero is in the uterus. And there's other terms as well, but those are some basic ones that, guys, if you don't get it from my test, you will absolutely 100% see these terms again if you're going to any medical field for people or animals. Get those terms down. If you don't get it from my class, if you think that it's, oh, well, I'm okay, I got it wrong this one time in microbiology. Man, if you can't figure out what in vivo or in vitro or in situ mean, it's your responsibility to know what those tests are going to be referring to when you're actually conducting these tests on your patients. Okay. So tuberculin skin test is in vivo because we're doing it on a living person's body, like while it's still on their living body. So it makes it in vivo. But the tuberculin skin test, we learned, especially when we just reviewed it in this previous chapter, that the tuberculin skin test relies on type 4 hypersensitivity, which is governed by your cytotoxic T cell memory. So if this will go, the tuberculin skin test is still immunologic because it relies on recognizing the antigen, which is tuberculin. The antigen is tuberculin. Does this rely on antibody? No. It relies on the antigen, tuberculin, being recognized by cytotoxic T cells. So this one doesn't have antibody involved, but it is still an immunologic test because it depends on the immune response. We're recognizing antigen. So the way I look at immunologic tests is that these are based on antibody and, well, whatever, and or antigen. You're recognizing that the antibody is there or recognizing that a specific antigen is there. And it is depending on immune reactions to get to that point. And that is an immunologic test. Oh my gosh, my M. Serology is essentially, for our intents and purposes, the term serology is interchangeable with immunologic. But serology originally came from serum. Serum is where you'll find antibody. Okay. So serology dealt with serum and then doing interactions based on immune response using serum. That's where the term came from. Nowadays, serology literally just means looking for antibody or antigen interactions based on immune response. Serology and immunologic essentially are interchangeable terms for that reason. For our purposes, for our class, that's definitely the case, okay? So indirect versus direct, these terms apply not just to ELISA, but to all immunologic tests. Indirect means you are asking the question, is an antibody present? If an antibody is present, it means that the person has seen the thing previously, has been sensitized to it at some point. We had time enough to develop antibody to the thing. And how strong that antibody response is an antibody titer. A titer is just a measure of the concentrations of the antibodies in the sample. We can use this to diagnose autoimmune diseases or previous infections or infections that we can't detect the virus, but we know that the person's been exposed to it previously, like HIV. Okay. Anyways, so sorry, I just got a message on my email and I got distracted. So, and that's measuring antibody. Direct tests are testing whether or not antigen is present. Antibody and antigen. So if your antigen is present, we're literally testing if essentially part of the pathogen is present. So it could be the pathogen is actually present. That's usually, but we're testing part of the pathogen, right? The antigen is the marker on the pathogen that your immune response is to. So that could be the spike protein on the surface or something. All right, so we're detecting if the antigen is there. In order to detect antibody, you will test that using antigen. In order to detect antigen, you will test that using antibody. You can view direct antibody interactions through either agglutination or precipitation. Agglutination happens when antibody is interacting with whole cells. Precipitation happens whenever antibody is interacting with antigen, not in the form of whole cells. So parts of pathogens. So just the spike proteins not attached to the virus or parts of the bacterial cell. So when an antibody interacts with the parts, that can lead to precipitation. When an antibody interacts with whole pathogen cells or whole viruses, that potentially could lead to agglutination. Whole pathogen, agglutination. Part of the pathogen, precipitation. Part, precipitation. Okay. Immunochromatography. This is using a laminar flow test, which is basically just a fancy way of saying fluid is going to go flow across a filter paper. Okay. That's laminar flow. So we are having lateral or laminar flow, flow of a sample across a filter paper. And when the sample touches the antibody line on the test, you'll see a color change. That is immunochromatography. It's immune-based. Chroma means color change, and graphy means test. It is an immune color change test. That's all that term means. And it usually refers to our lateral flow rapid tests like your home COVID test or your home flu test or pregnancy tests. The antibody titer. We already mentioned that one's dealing with the concentration of the antibody, and it can relate to how recently you're exposed to an antigen. Specificity and sensitivity. How specific a sample is means that we are only recognizing the, I have got to mute this. This is getting annoying. Okay. Antibody titer. God, I got sidetracked again. Specificity. If a test is specific, it means we are only recognizing actual positives and not things that kind of look like the positive. So if you call something that kind of looks like flu, but isn't flu, and you're saying it's positive for flu, that is a false positive. So specificity, it's specific to this exact specific antigen, not things that look kind of like it. That's specificity. Only that specific thing we will call positive, not things that are kind of like it. So the more specific a test is, the less likely a false positive is. For sensitivity, you want to pick up on every single person that could be positive. Even if they only have one virus in their blood, we want to call them positive because they have the virus, they are positive. So sensitivity, we want to avoid calling a test negative or a person negative whenever they might actually have tiny amounts that makes them positive. So the more sensitive a test is, the more likely you'll be able to say that they are positive, even if they have just tiny, tiny, tiny amounts of positive. If you call somebody negative, but you just tested this vial of blood didn't have virus in it, but maybe somewhere in the rest of their body there is a virus, right? So if you called that person negative, that would be a false negative. They are positive. You just didn't test them positive in your test. The more sensitive a test is, the less likely a false negative is. All right, in vitro serum testing. We can take serum out of somebody's body and do tests with the antibody antigen response. A Western blot processes proteins in a gel very much like gel electrophoresis with DNA. So we'll spread out the proteins. They'll move to the other end of the gel based on how big they are. So larger ones move slower, smaller ones move faster. We'll separate the proteins out into these bands, and then we will use an antibody to see if the antigen is in the gel. Or we can take the serum and spread the serum out and use an antigen to see if the antibody is in the gel. We can do either end of it. But Western blot is separating out proteins, pushing the proteins onto a special filter paper, and then looking at antibody antigen interactions on that filter paper. Immunofluorescence is labeling antibodies with a fluorescent molecule so that we can see if they bind to a sample or not, and by looking at them, if they fluoresce when we shine a laser at them. So it's using antibody antigen interactions and looking for the actual visible fluorescence. ELISA, we did that in lab. It's enzyme linked immunosorbent assay. Is always going to be done in those 96 well plates. And it is highly specific and sensitive. And it's going to depend on antibody antigen interaction. And we're going to quantify that because an enzyme that is linked, right, to a secondary antibody. The enzyme is linked to a secondary antibody, and the enzyme is cleaving a substrate. And when the enzyme cleaves a substrate, we get the color change. So the fact that the enzyme is linked to an immune molecule, and the enzyme that is linked to the immune molecule cleaves the substrate and causes a color change, that's what makes it ELISA. The fact that you have an enzyme that is linked makes it ELISA, not the color change. The fact that the enzyme that is linked to the immune molecule leads to the color change makes it ELISA. We also have complement tests. This just relies on whether or not, when we mix samples with red blood cells, if they cause the red blood cells to lyse. And that is going to be depending on complement causing the blood cells to lyse or the complement being used up by antigen, preventing blood cells from lysing. I'm not going to get into any more detail than the absolute basics of complement assay for the immunologic test, because this gets really confusing for people understanding how they you first have to understand the complement cascade and how it functions. So when I ask questions on the test about the complement, if I ask a question about the complement test on the exam in regards to the immunologic assay that we use based on complement, I will just ask that, is there a test that relies on complement for immunologic testing? It'll be like that basic of knowledge, because that's all we're going to talk about. Okay. And we already talked about the tuberculin skin test. All right, moving on to the genetic tests. A test is only a genotypic or genetic test if it is actually based on nucleic acid sequence, not the products of the gene. Products of the gene and the expression of the gene is a phenotypic trait. If we're looking at if we're depending on the actual genetic sequence, that's what makes it genotypic. And so that includes PCR, nucleic acid hybridization, including FISH, which is fluorescent in situ hybridization. This is taking a known sequence of DNA and labeling it with a fluorescent probe. And this DNA sequence matches up this mutation. If I mix that mutated sequence that's labeled with fluorescence with the cells, and then your cell sample has an actual fluorescence happen, it means that your genes have that mutation. And so we can use that without having to sequence your DNA or rely on PCR and just seeing if the probe matches your sequence. We can also do this for bacterial sequences to see if your patient sample has streptococcal bacterial gene sequence hybridizing with it. And then microarrays, where you have a whole like tray or plate or chip that has a whole bunch of different samples on it. We can mix your patient sample on there, see which spots your patient sample matched onto, and that can help with diagnosing infections as well as mutations for cancers and things like that. Mass spectrometry relies on the mass of your particles. So we will shoot a laser at your patient sample and ions will fly off. And based on their actual ion charge, as well as the mass of the particles, they'll reach the detector at different times. And when they reach the detector, that is going to determine their time of flight, the time it took them to get to the detector. And we can actually break down molecular makeup of samples based on those results. So this has to do with the molecular makeup of things. And then imaging, these are pretty straightforward. Most people know about imaging. We are looking for areas of infection or areas where there are visible tumor or abnormal aspects of your patient's anatomy or whatever, to look at that with imaging without having to use invasive surgical techniques to investigate that stuff. So let's get in finally to our questions for most missed topics. I wanted to take some time on that explanation because I feel like we didn't cover it well in our lecture. So we'll start with our hypersensitivity stuff. This is a slide that goes over the hypersensitivity stuff just a little differently. I'm not going to go over this slide, but this is here for your review if you need help with that. So our questions to review this stuff: which of the following is the most immediate and effective treatment for anaphylaxis? So we know anaphylaxis is a systemic response. This happens through the bloodstream. Anaphylaxis can cause constriction of the airways and reduce blood flow to the extremities. This is a type 1 hypersensitivity reaction. And the best treatment for anaphylaxis, because it can cause a very quick response within minutes of exposure to the allergen, and it is systemic, that the best way to reverse those effects is through epinephrine injection. This will open up your airways, as well as increasing blood flow to like the brain, for example. In the sensitization phase of a type 1 hypersensitivity reaction, what role do IgE antibodies play? So what happens in the type 1 hypersensitivity is that first you will see your, first you will see your antigen, and then the antigen will essentially trigger your B cell to produce IgE, and then the IgE will then add on to a mast cell, and then that will degranulate when we see the allergen again. So let's see what we can look through with our choices. Will IgE neutralize the antigen in the bloodstream? No, it's going to not directly do that. It's going to attach to the mast cell and then cause the mast cell to degranulate. So that's not our answer. Will the IgE bind to allergen-specific receptors on mast cells and basophils? This is probably going to be our choice, that allergen, after we have created IgE, the IgE will be on special receptors on mast cells and basophils. We just didn't cover that in our lecture. And that these will degranulate after we see the allergen again. But let's look through our other choices. Does the IgE sensitization lead to complement protein destroying the allergen? No, leads to mast cell degranulation. Or will IgE sensitization cause phagocytosis of the allergen by the macrophages? No, we are having degranulation of the mast cell and the basophil. So our answer is B. So hemolytic disease of the newborn and the effects of that are covered here. So if you want more detail on how hemolytic disease of the newborn occurs, why death can be a common problem, or why you have very serious effects if you do still survive this sort of a situation, this is the issue of symptoms that can be caused by hemolytic disease of the newborn if the newborn were to survive, or during the time that the newborn might be surviving leading up to death. So jaundice, anemia, loss of red blood cells because we're attacking them through the complement pathway, enlargement of the liver and the spleen due to dealing with the toxic components of the hemoglobin and the damaged red blood cells. And then hydrops fetalis is a fluid buildup in fetal tissues due to the inflammatory response caused by the damaged red blood cells. Hemolytic disease of the newborn can usually be caused by any blood type discrepancy, so any rejection of blood type. It could be due to RH or ABO compatibility, but it is more severe with RH, and that's why we're focusing on that. You can survive typically ABO incompatibility, but you won't typically survive RH incompatibility. Additionally, RH incompatibility leading to hemolytic disease of the newborn is more common than ABO incompatibility. So there's that. We have our memory thing here. Helpful rhinos always share pink babies. So helpful hemolytic disease of the newborn, rhinos for the RH factor, and then always leading to like anemia, and ABO blood group types. Sharing is going to be the sensitization, and then pink will be the pathogenesis aspects of that, where we're going to be having the symptoms of the disease, and then the babies is dealing with the I guess that was the blood group one. I have the notes on this one as well at the bottom of the slide when you look at the notes for the slides yourselves, if you care about those little mnemonics to help you remember that. But anyways, moving on. Hemolytic disease of the newborn for RH compatibility is going to be the RH negative mother attacking an RH positive baby, and there has to be a sensitization for it. So our question is, which of the following is a primary method to prevent hemolytic disease of the newborn due to RH incompatibility? What do we do to prevent that? We administer an immune globulin that reacts to the antibodies made by our RH negative mothers. So RH negative mothers make antibodies against the RH group in the baby. And so we have made RhoGAM that blocks those antibodies. We don't do blood transfusions during pregnancy. We don't want to replace the mother's blood with positive blood because the mother's body is going to attack the positive blood. We don't provide vitamin K to the newborn. The vitamin K isn't going to have anything to do with the mother's response and immune response to the baby. And we don't provide antiviral medications during pregnancy because viruses aren't what is causing this issue. So it's going to be B, RhoGAM. So our second question, a newborn is diagnosed with severe jaundice, anemia, and an enlarged liver, which are indications of hemolytic disease of the newborn. The mother's blood type is AB negative and the baby's blood type is A positive. What is the most likely cause of these symptoms? Well, if the mother was AB negative, her body made anti-RH positive antibodies that could attack the baby's RH positive blood. So this is likely due to RH incompatibility. How do we know that it's not ABO incompatibility? Because the mother is AB, the baby is A. To the mother, the A looks normal. It's the RH that's the problem, not the ABO. There's no indication that there is a viral issue going on here, but there is an RH incompatibility. And there's no indication that diabetes is playing a fact. This is going to be an RH incompatibility, for this is the most likely cause of these symptoms. All right, for tissue grafts, these are the different versions of the grafts. Remember, autograft is self-to-self. Allograft is from people who are not genetically identical. Xenograft is from a non-human species to a human. And then isograft is going to be from genetically identical people. So let's go ahead and go into the questions for this. Which type of tissue graft is most likely to require lifelong immunosuppressive therapy to prevent rejection? Which of these is going to have the most difference here. So autograft is self to self. We should not have a problem with self things. Isograft, these are identical from an identical twin. We shouldn't be rejecting that. These are from different people, but at least they're people to people. This is from animal to people. This is where the biggest risk is going to be. This is where the biggest difference is. You're still going to need immunosuppression for the allograft, potentially. But for animal to people, those are massive differences. So if you had an organ tissue graft from an animal to a person, the immunosuppression for maintaining something like that is going to be a greater concern. So xenograft is our answer for that one. That's the biggest difference between the donor and the recipient in those examples. So next question, a patient receives a heart valve transplant from a pig. So this is a xenograft, right? Based on our knowledge. I haven't even gotten to the choices left. I'm looking down there and it's basically that's what the question is. What type of tissue graft is this, and what is a major concern associated with it? Well, we've already determined that it's xenograft, so we'll just go right down to that one. It's not autograft because this is self. It's not isograft because it's not genetically identical. Iso, identical, right? Allograft is from a different person. You would say allo to a different person, maybe. I don't know. I'm reaching here, but it's something. This would require immunosuppressive drugs, but this is from an animal to a human. This is a xenograft. So there's a high risk of rejection and potential disease transmission from animals to humans with those zoonotic diseases that can be transmitted from animals to people. So finally, getting into chapter 18, our most missed concepts here are the differences between our phenotypic, immunologic, and genetic tests. And this was definitely the case with a lot of people on the quizzes, not understanding that if a test is a differential media, it is a media that tests for metabolic differences. So that's what makes it differential media. But the fact that it tests for metabolic differences makes it a biochemical test. And the fact that it is a biochemical test makes it a phenotypic test. Those subcategorization steps were definitely something that people were missing on the quiz, and that tends to carry for the exam as well. So this is about understanding our different groups here. Remember that immunologic is antibody or antigen, and it's related to the immune response cells. Genetic testing. This is relating to actual nucleotide sequences. So nucleic acid sequence. If we're doing anything related to the actual nucleic acid sequence, that's the genetic or genotypic test. Phenotypic tests are going to be based on identifiable traits. If we learned about the difference between genetic or genotype versus phenotype, this is literally that. Genotype is looking at the actual genetic sequence. Phenotype is the trait that is expressed. So when you're looking at metabolic products, you're not looking at the genetics that led to the metabolic products. You're looking to the metabolic products. Those are measurable traits. Those are effects of what is expressed. That's the difference there. So that's the big picture of what phenotypic really means. Phenotypic tests are going to include biochemical tests that are going to be related to metabolism, or phage typing that has to do with the phenotype of that bacterial species being able to be infected by that virus, or Kirby-Bauer. Kirby-Bauer is not immunologic. A lot of people are getting confused with that. Your body didn't make the antibody. We got a drug outside of that, and we're looking at the phenotype of your infectious pathogen. Whether or not your infectious pathogen is susceptible to this antibiotic is a phenotype. So those are phenotypic tests. When we do stains, gram stains, endospore stains, acid-fast stains, if your organism stained positive for acid-fast, you have mycolic acid, that's a phenotype. That is an actual expression of that trait. That is a feature that that organism has that we're testing. Okay, so phenotype is dealing with the traits of your pathogen. Immunologic is dealing with immune responses to a pathogen, and genotypic is dealing with actual genetic sequences. So let's write that out to the side. Phenotypic. These are expressed traits of a pathogen. Immunologic. This is dealing with antibody or antigen immune response factors. And then genotypic. This is just dealing with nucleotide sequence. So our question is, which type of diagnostic test will be most appropriate for identifying a bacterial pathogen based on its genetic material? Okay, so we know it's a genotypic test. We know we're looking at nucleic acid sequence. So which of these tests look at nucleic acid sequences? Gram staining is a phenotypic. ELISA is an immuno. It's relying on immune reactions. Polymerase chain reaction is a genotypic. We are looking at the actual genetic sequence. Biochemical test strip. This is a phenotypic. Next question. A patient undergoes a test to determine the presence of antibodies against HIV. Well, we're looking at antibodies. Which category did that fall under? Immunologic. Jeez. All right. Phenotypic would be looking at biochemical traits, or whether it's stained positive or negative, or the phage typing, etc. Genetic test looking at the actual specific genotypic makeup, the nucleic acid makeup, the ATCGs of it. And then biochemical tests are a type of phenotypic test. All right, then we have sensitivity versus specificity. Sensitivity. So if you guys want a little more explanation on these, you can go over these. Or remember, sensitivity is the ability to test true positives and identifying all of the true positives that are there. Specificity is the ability to test only true positives and making sure that negatives are true negatives. So let's look at our breakdown over here. I'm going to write our little sheet summary on the side. Sensitivity. We're going to find as many high number of positives that are truly positive and not miss any of the positives. That means no false negatives. Specificity is dealing with only true positives, no false positives. All right, so our first question. A diagnostic test has a sensitivity of 95%. What does that mean? Does this mean that the test correctly identifies 95% of patients without the disease? No, this means that if it is sensitive, it will get 95%. What this is actually saying, so we'll go ahead and analyze this question just to break it down before we get to our choices. What this is actually saying is that if you had 100 patients with the disease, its sensitivity says we'll catch 95% of those. That's what that means. So there might be 5 people we wouldn't call positive. We call them negative. That's a false negative. All right, so that's the actual measure of the sensitivity. So does this mean that the test correctly identifies 95% of patients with the disease? This is probably our answer. Let's look at the other ones. Does it say that the test has a 95% chance of being positive in healthy people? No, it means that of all 106 people, 100 sick people, we will say that of those 100 that are sick, this test will be able to correctly say 95 of them are sick. The 5 of them that it says they are not sick are sick. We just didn't catch them. That's what's going on here. And so D says the test has a 95% chance of correctly diagnosing the disease when it is absent. How is it correct if you're saying that doesn't make any sense? So this one, literally the logic is gone. That's why B is our answer. This is correctly identifying 95% of patients with the disease. It is saying we will call 5% of patients that have the disease negative. 95% of them will be called positive. That's the measure of sensitivity. The next question, a new test for tuberculosis is designed to have high specificity. So that's this one down here. What is the primary benefit of this characteristic? So if it's highly specific, we should be calling things that are positive, positive, but not things that look like the positive, positive. So this is avoiding false negatives, okay? So will the test detect a high percentage of people with tuberculosis? This is dealing more with sensitivity, but we'll come back to that. We'll look at our other options. The test will accurately confirm that a person does not have TB if the test result is negative. So this is pretty close to what we're getting at here. We're saying that if we're calling you negative, that means that you are negative. And if we're calling you positive, you are positive. And there shouldn't be any crossover there. That's what specificity deals with. The test will have a high rate of false negatives. No, the goal is to have no false negatives. The test will identify a high number of non-TB infections. No, if this test is specific for tuberculosis, we are only identifying whether you have tuberculosis or don't have tuberculosis. So B is our answer for this. The test will detect a high percentage of people with TB isn't really what the specificity is testing. That would be sensitivity. Specificity is specifically saying we are identifying TB as positive and not TB as negative and not having any gray area there. And that's why specificity, the answer would be B. Oh, okay. We made it through it. So that is the end of our week 12 review, and I will update the unit 3 questions as well. All right. Yes. Can I go over the questions for last week review that are on the slides? So yes. So let me pull up last week's review really quickly. For those of you who are still hanging out with me, I don't mind going over that. Nope, that's not what I meant to open. I gotta find it first is the problem. So weekly reviews. That was week 11. Maybe. There it goes. Okay, so I have to download this first because this is my iPad. I don't have it on here automatically. Save to file. Lecture. Save. Okay. No, come on. No, I wanted week 11. Okay. Files. Week 11. That's what I want. Okay, here we go. Okay, so let's go over. We did our review on the concepts, so I'm going to jump straight to the questions if that's okay with you, Heather. So I can see your comments. I'm going to leave the chat up on the side over here, but still go over these questions. Okay, so our first question is, which of the following is a primary function of mucus in the respiratory tract? And so mucus is there as we have in our immune system, we have the 3 levels of our immunity. We have first line of defense, which is like our physical barriers, our second line of defense, which is our innate response, and our third line of defense, which is our acquired immunity. The first line of defense, our physical barriers and our chemical barriers, this would be falling into that. So mucus is literally like sticky, so it's there to trap pathogens in the lungs. So you're exactly right. That's going to be B. That's an easy one. Our next question, which of the following conditions might compromise the first line of defense, increasing the risk of infection? So our first line of defense, again, is our chemical and physical barriers. So would exercise and a balanced diet put that at risk? I would hope not, based on what everybody's telling me, right? Yes, and so we think B is going to be the answer for this one. Chronic use of corticosteroids leading to thinning of the skin. So corticosteroids, steroids in general will reduce your immune function, but your immune function is actually part of like your second and third line of defense. But corticosteroids can also lead to thinning of the skin. And this puts the skin at risk for being broken and leading to a loss of a barrier function there. All right, so let's go on to the next ones. Which of the following is a benefit of fever in the context of an infection? So we have plenty of benefits of fever. We have benefits that help our body function better, and we have benefits of fever that inhibit the microbes from growing. So let's look through our choices. Which of the following is a benefit of fever in the context of an infection? Fever decreases production of white blood cells, reducing inflammation. That is not true. Fever increases the function of our white blood cells and can actually lead to increasing inflammation. But remember that inflammation is important for fighting pathogens. So that's actually a good thing. Or B, fever enhances the ability of the immune cells and speeds up the healing process. Fever does do this for us. That's part of one of the benefits it does for us. Fever increases the ability of iron, the availability of iron in the bloodstream. Okay, so fever actually eliminates iron from the bloodstream and hoards it away. The pathogens do use it for growth. So we remove iron from the bloodstream so the pathogens can't use it. That's part of fever response. Next, fever decreases the body's metabolic rate, slowing down our immune responses. Fever increases our metabolic rate. So our answer is B here again. Our next question, in a clinical setting, why might a fever be a significant indicator for a healthcare provider? So fever, if it is present, indicates that there's probably infection. There's at least an inflammatory response going on to something foreign. So let's look at our choices here. Does fever suggest that a patient has a cold, which might require immediate antiviral treatment? I don't know. Let's look at our other choices. Does fever indicate the presence of an infection or inflammatory condition that may need further investigation? Fever is a general indicator of something going wrong. Fever doesn't suggest a cold specifically, right? So that's the difference here. It indicates the presence that there might be an infection or an inflammatory condition, but we should figure out what's going on with that. That's why B is probably our right answer. All right, so I do have these down here in the notes, so I'll have that up for a second so we can look at that, but I'll open that if we need it. But C, confirms that the patient is dehydrated. No, fever doesn't mean you're dehydrated. If you aren't drinking fluids, you might be. So it's definitely not that. And then guarantees that the patient has a bacterial infection rather than a viral one. You can absolutely have fever with viral infections, and we know this because COVID, because flu. And so that's not an indicator of bacterial or viral. So our answer is B. That's the best answer. Could the patient have a cold and then have a fever because of the cold? Yes. But we don't know that just because of the fever. So that's why B is the answer here. Our next questions. Which of the following correctly describes the initiation of the lectin pathway of complement activation? There are 3 pathways for complement activation. The classical, which is governed by antibody, the lectin, which is when lectins in our body will identify microbe sugars. These are sugars that are associated specifically with microbes. And then we have the alternative pathway, which associated is going to be able to recognize molecular patterns like LPS. Very similar to the concept of PAMPs. So the lectin pathway of the complement activation will be, according to our notes over here, identify basically microbial sugars on the surfaces of bacteria. So our choices are, is this a pathway that is initiated by antibodies? That is the classical pathway. Is it the pathway that is triggered by the presence of LPS? That's the alternative pathway. It is the pathway that begins with mannose binding lectin. Well, I mean, binds to a specific carbohydrate on the pathogen surface. Or is it the pathway that starts when a complement complex binds to a pathogen surface directly without antibody binding? This isn't what's happening. It's just confusing word salad. Okay. Next question. In a clinical scenario, a patient with recurrent infections is found to have a deficiency in C1q, which is a part of the complement system. So they have a complement deficiency. This is a particular part of complement system where we recognize antibody. The part of the complement system that recognizes antibody is the classical pathway. So which complement pathway is affected? Not the lectin pathway, because that recognizes lectin binds to sugars on microbe surfaces. It's not that one. It's not the alternative pathway that's governed by LPS. It's going to be the classical pathway specifically, because that's the one that is governed by antibody there. All right. Next questions for 16. Which step in the complement cascade involves, this is supposed to be 15. It says 16 on that other slide, but this is 15 still, sorry. For last question for 15. Each step in the complement cascade involves the assembly of activated complement protein molecules to form a structure that creates pores in the pathogen membrane. So in complement, our functions of complement can be for opsonization, or providing molecular handles for phagocytosis, or it could be for activating the inflammatory response, or it could be for creating the MAC, which is the membrane attack complex. Right. And so that's the big picture of it, right? So when we are doing our membrane attack complex, complement function, in order to get to membrane attack complex, we first have to initiate, then amplify, then polymerize into the MAC. So we have our notes down here that this is going to be C because we're developing the MAC. The MAC is developed with the polymerization step. So you're not wrong. And the exam questions won't be this vague. I won't say vague, because to me, this is getting still at the point of making the membrane attack complex. We're getting to making the membrane attack complex, but the membrane attack complex isn't made from the initiation step. It isn't made from amplification step. We're building the membrane attack complex when we're polymerizing. Okay, so that's how we're getting to that answer there. Okay, the next question, does that make sense, at least how I got there? So, right. So what happens is you will, in the steps, so the 4 steps of complement activation are initiation. Well, I guess this is basically 3 steps, but the 4th step is membrane attack complex. But in order to make the membrane attack complex, you have to polymerize. So polymerize is basically initiation is starting the complement cascade. You have triggered this protein. This protein will then trigger all of these proteins. And then these proteins will trigger these proteins, and this one will trigger these proteins, and this triggers these proteins, etc. So this is the initiation. This process is the amplification. And then once you get all of these guys, they can come together. Once you've amplified them, they will come together and polymerize. That's the polymerization step. The end result of the polymerization after you've polymerized is the membrane attack complex, but you cannot form the membrane attack complex without polymerization. Yeah, is that making a little more sense how we got there? So that's why that is the answer for that. All right, so the next question is, a patient is diagnosed with a condition that impairs the formation of the membrane attack complex. We form the membrane attack complex through the polymerization. That's how we get that result. So once this has polymerized, then you get the MAC. All right, so polymerization is the process of making the MAC. And remember, polymerization means the same thing no matter what concept we're talking about. So polymerization, like PCR, it means polymerase chain reaction. So polymerize means to bring small molecules together to build a more bigger molecule out of the small molecules. That's always what it'll mean. And so our bigger thing that we're making from our small things is the membrane attack complex. All right, so if we're affecting the formation of the membrane attack complex, which would be most directly affected, it's not the initiation, because that's initiating all of complement. It's not the amplification, but it's going to be that polymerization step there again, because that polymerization, sometimes I write extra things and I didn't write it this time, but it's that polymerization step that is actually, oh, I have an eraser now. I was wondering why things were disappearing. It's that polymerization step that is going to be leading to developing that membrane attack complex. All right, so we're going to move on to chapter 16 now. The questions for chapter 16. Which type of MHC molecule is responsible for presenting antigens from intracellular pathogens to CD8 positive T cells? So if it's intracellular, just to summarize that one, remember that MHC1 looks like I to me, so that's inside. That means MHC2 will present stuff from out. All right, the next question. A patient with a genetic defect that affects the expression of MHC2 molecules, this identifies outside stuff, will have difficulty with which of the following? Will they have a problem presenting endogenous? No, because this is MHC1 related. Will they have a problem with activating helper T cells? Helper T cells recognize MHC2, right? And so that's not an issue with targeting inside stuff with virus, and not eliminating cancer cells. That's all inside stuff. All right, moving on to the next thing. We have a question here. Antibody class switching. So our antibody and our antibody classes. We have 5 types of antibody immunoglobulin. IG, IG, IG. We have IgD, which is just our receptor on our B cells. IgG, which is in our blood. It's our workhorse. It's our big daddy. We have IgA, which is in the mucus. We have IgM, which is the first one. And we have IgE, which is for parasites and allergies. all of these, oh no, what happened? I accidentally touched the eraser. All of these, this is determined by the FC constant region. The FAB is the actual antigen associated area. That's the variable region of the antibody. Okay, so during antibody class switching, so if we're making this one first, and then we're switching to these guys, either these two or this one. This one doesn't count, or you'll switch to this one. It's on the receptor. So during antibody class switching, what change occurs in the antibody molecule? So if we're switching from M to G, A, or E, this is the constant region. Right. So yes, so switching involves the heavy chains. Exactly. So the heavy chain is where the constant region is. The heavy chain at the bottom of the heavy chain is the constant region. At the top of the heavy chain, you have part of the variable region, but the other part of the variable region is on the light chain. But the heavy chain, the base of it is where the constant region is. So remember, your antibody looks like this. This is your heavy chain. This is your light. This is your constant. This is your variable. All right. So moving on. Patient is diagnosed with an immunodeficiency that affects their ability to switch antibody classes from IgM to IgG. And IgG is our workhorse, and it's really important in our proper memory and protection. Let me zoom in on this a little bit. Okay, so which of the following would likely be true about their immune response? The patient would have a normal IgG levels. No, if they can't, they have to start out with IgM, and then you switch to IgG. So you would have the opposite of that. The patient would have difficulty producing effective long-term immune responses. Yes, the IgG is what's important for the memory and for the proper response in our memory response later during the amnesiatic response. The patient would have high levels of IgE. This has nothing to do with either of those. Or the patient would have an overproduction of IgA. Again, nothing to do with those. You start from IgM always. And then you can switch to IgD. Oh, sorry, IgA, IgE, or IgG. And you always had the B cell that has the receptor on it. This is IgD. How many more we got? Okay, just 2 more sets. Okay, moving on to our T cell guys. Which helper T cell subset? So you always start out as a CD4 T helper, just general. This is what we call naive. Once we activate, then these guys will switch to Th1 and Th2 helper cells. So which type of T helper subset, which of these guys here, is involved in promoting antibody production? That's going to be your Th2 response. That governs your outside response. So that will govern everything dealing with antibodies. So it governs outside. Governs are captains. All right, a patient with an autoimmune disease has elevated levels of pro-inflammatory cytokines, and that reduces the function of the T regulatory cells. So this person is losing regulation. They're having too much inflammation. So which of the following is most likely affected in this patient? Do we have going on here the ability to activate? We have a problem with activating our macrophages and cytotoxic T cells. Do we have a problem with regulating the immune response to prevent autoimmune damage? Yes, we've got too much inflammation going on. We're regulating things that shouldn't be regulated. This is the biggest risk here. Do we have promotion of B cell antibody production, or do we have suppression of the inflammatory response? Well, obviously, we don't have suppression of anything. That can't be that. It's not affecting our ability to activate macrophages and cytotoxic T cells. We actually have overactivation. So we don't have, this is talking about underreaction. And promoting B cell antibody production and allergic responses. The T regulatory cells don't regulate antibody responses. They regulate the T cell responses, and that these T cells can be regulating activation of other cells that could be autoimmune. And so that's why that's going to be that answer. Our next question, which of the following molecules is released by cytotoxic T cells to induce apoptosis? These are going to be our perforin and our gram signs. Remember, our perforin pokes holes, and our gram signs are going to digest what's in the cell. And when this happens, this causes the attacked self cells to go through cell suicide. That's called apoptosis. All right, our next question. A patient is experiencing a viral infection and is found to have a deficiency in cytotoxic T cell function. So the cytotoxic T cells were found to not being able to attack infected self cells. All right, can't attack infected self. So which of the following is most likely to be directly affected by this? Will we have a problem with activation of T helper cells? Will we have a problem with producing memory B cells? Will we have a problem with phagocytosis of extracellular bacteria by macrophages? Or will we have a problem with our ability to kill cells that are infected with intracellular pathogens? So your cytotoxic T cell response is responsible for killing infected self cells. That means D is the answer for this one. All right. So we've gone through the questions from chapters 15 and 16. We've also covered 17 and 18. I will update Unit 3 to make sure that it's covering things at the level that I expect you guys to understand it where I can. I can't put like every single thing for every single topic ever in there, but I'll do what I can to put what I can in there. Do you have any questions? Either Heather or anybody else that might be joining us. Do you guys have any questions? All right, if that is it, I am going to go ahead and, yeah, it's a lot. 15 and 16 are definitely our biggest, heaviest chapters here. And I feel like understanding 15 and 16 takes you a really far way with 17. And so, and then 18, the stuff, the tests in there, many of them we've already learned about. It's just categorizing them. And then I feel like 11 and 12, it's about flashcards with 11 and 12. 11 and 12 are flashcard chapters. So hopefully focusing a lot more energy on 15 and 16. And then trust me when I tell you, like, this stuff will help go a long way with understanding infections in your patients and how everything is operating in their immune system. Very under taught. And I wish I had more time to cover that stuff with you guys, but it is what it is. So if you guys don't have any more questions other than that, I'll go ahead and end the review. Yeah, 11 and 12 is definitely definitions. Yeah, fingers crossed for you guys. If you guys get to studying and you're still having problems, feel free to email me and do what you can for understanding that content. Explain them to each other. You know, imagine that you're explaining it to your friend or somebody and stuff like that, and that'll help go along with trying to work on understanding, and it'll help show some areas maybe where you need to work on more for studying as well. So good luck studying, everybody, and feel free to email me if you have any questions. And other than that, I'm going to go ahead and call it a day.